Welcome to Module 1.1.2, the second module in Unit 1, Module 1.1. In this module, we'll discuss the pituitary gland, the hypothalamus, and several hormones and structures related to them. So just as a recap, rewatch the last 3 to 5 minutes of the previous lecture video where we covered the hypothalamic response in an example, because the next two slides offer kind of a refresher of that information that was at the tail end of the last video. But in this slide, the hypothalamus is defined and explained. And on this slide, it recaps the hypothalamus-pituitary complex. Also, be sure to familiarize yourself with the learning objectives for the module here. Remember that both the quiz and the test questions are written based off of the objectives. All right, getting into it. So, figure 18.6 here, or 18-6, shows the anatomy of the pituitary gland, or the hypophysis. This small oval gland lies nestled within the sella turica, which is a depression in the sphenoid bone. The pituitary gland is inferior to the hypothalamus, and it's connected by the slender funnel-shaped structure called the infundibulum. The base of the infundibulum lies between the optic chiasm and the mammillary bodies. Cradled beneath the sella turica, the pituitary gland is held in position by the sellar diaphragm, which is a dural sheath that encircles the infundibulum. The sellar diaphragm locks the pituitary gland in position and isolates it from the cranial cavity. The pituitary gland has an anterior and a posterior lobes that differ in function and developmental anatomy, which we'll talk about a little later. And it releases 9 important peptide hormones, which we will also discuss in detail. 7 come from the anterior lobe and 2 from the posterior lobe. All 9 hormones bind to the membrane receptors, and all 9 use CAMP as a second messenger. The anterior lobe of the pituitary gland is also called the adenohypophysis, contains a variety of endocrine cells. The anterior lobe has 3 regions. 1, the pars distalis, which is the largest and most anterior portion of the pituitary gland. 2, an extension called the pars tuberalis, which wraps around the adjacent portion of the infundibulum. And 3, the slender pars intermedia, which is a narrow band bordering the posterior lobe. An extensive capillary network radiates throughout these regions, giving every endocrine cell immediate access to the bloodstream. The hypothalamus controls the production of hormones in the anterior lobe of the pituitary gland by secreting specific regulatory hormones. At the median eminence, a swelling near the attachment of the infundibulum, hypothalamic neurons release regulatory factors into the surrounding interstitial fluids. Their secretions enter the bloodstream quite easily because the endothelial cells lining the capillaries in this region are unusually permeable. These fenestrated, which fenestrated means window, capillaries allow relatively large molecules to enter or leave the bloodstream. The capillary networks in the median eminence are supplied by the superior hypophyseal artery. Before leaving the hypothalamus, the capillary networks unite to form a series of larger vessels that spiral around the infundibulum to reach the anterior lobe. In the anterior lobe, these vessels form a second capillary network that branches among the endocrine cells. This vascular arrangement is unusual because it carries blood from one capillary network to another. In contrast, a typical artery conducts blood from the heart to a capillary network, and typical vein carries blood from a capillary network back to the heart. Blood vessels that link two capillary networks are called portal vessels. In this case, they have the histological structures of veins, so they are also called portal veins. The entire complex is a portal system. Portal systems are named for their destination, and this particular network is the hypophyseal portal system. Portal systems are an efficient means of chemical communication. This one ensures that all of the hypothalamic hormones entering the portal vessels reach the target cells in the anterior lobe before being diluted through mixing with the general circulation. The communication is strictly one way. However, any chemicals released by the cells downstream must do a complete circuit of the cardiovascular system before they reach the capillaries of the portal system. 2 classes of hypothalamic regulatory hormones exist: releasing hormones and inhibiting hormones. A releasing hormone, or Rh, stimulates the synthesis and secretion of one or more hormones at the anterior lobe. In contrast, an inhibiting hormone prevents the synthesis and secretion of hormones from the anterior lobe. Releasing hormones, inhibiting hormones, or some combination of the two may control an endocrine cell in the anterior lobe. The regulatory hormones released at the hypothalamus travel directly to the anterior lobe by the hypophyseal portal system. Negative feedback controls the rate at which the hypothalamus secretes regulatory hormones. The primary regulatory patterns are diagrammed in Figure 18-8 that you see here, and we'll refer to them as we examine specific pituitary hormones. There are several hormones of the anterior lobe. The functions and control mechanisms of 7 hormones from the anterior lobe are reasonably well understood. Thyroid-stimulating hormone, adrenocorticotropic hormone, 2 gonadotropins called follicle-stimulating hormone and luteinizing hormone, prolactin, growth hormone, and melanocyte-stimulating hormone. Of the 6 hormones produced by the pars distilis, 4 regulate the production of hormones by other endocrine glands. The names of these hormones indicate their activities, but many of the phases are so long that we often use abbreviations instead. The hormones of the anterior lobe are also called tropic hormones, trope to mean a turning, because they turn on endocrine glands or support the functions of other glands. So let's go through several of these. Let's start off with thyroid-stimulating hormone. So the thyroid-stimulating hormone, or TSH, or tyrotropin, targets the thyroid glands and triggers the release of thyroid hormones. TSH is released in response to thyrotropin-releasing hormone, or TRH, from the hypothalamus. Then, as a circulating concentration of thyroid hormones rise, the rates of TRH and TSH production decline. Adrenocorticotropic hormone, or ACTH, also known as corticotropin, stimulates the release of steroid hormones by the adrenal cortex, the outer portion of the adrenal gland. ACTH specifically targets cells that produce glucocorticoids, hormones that affect glucose metabolism. ACTH release occurs under the stimulation of corticotropin-releasing hormone, or CRH, from the hypothalamus. As glucocorticoid levels increase, the rates of CRH release and ACTH release decline. Gonadotropins. The hormones called gonadotropins regulate the activities of the gonads. These organs, the testes in the males and the ovaries in the females, produce reproductive cells as well as hormones. Gonadotropin-releasing hormone, or GnRH, from the hypothalamus stimulates the production of gonadotropins. An abnormally low production of gonadotropins results in hypogonadism. Children with this condition do not mature sexually, and adults with hypogonadism cannot produce functional sperm in males or oocytes in females. The 2 gonadotropins are follicle-stimulating hormone and luteinizing hormone. And we'll get into those a little bit more in just a second, but we'll also talk much more about these 2 hormones when we get into Module 2. So follicle-stimulating hormone, or FSH, or pholotropin, promotes follicle development in females, and in combination with luteinizing hormone, stimulates the secretion of estrogens by ovarian cells. Estradiol is the most important estrogen. And in males, FSH stimulates nurse cells, which are specialized cells in seminiferous tubules where sperm differentiate. In response, the nurse cells promote the physical maturation of developing sperm. FSH production is inhibited by inhibin, which is a peptide hormone released by cells in the testes and the ovaries. Luteinizing hormone, or lutropin, induces ovulation, the production of reproductive cells in females. It also promotes ovarian secretion of estrogens and progesterone, which prepare the body for possible pregnancy. In males, this gonadotropin is sometimes called interstitial cell stimulating hormone, or ICSH, because it stimulates the production of sex hormones by the interstitial cells of the testes. These male sex hormones are called androgens, andros to mean man, and the most important one is testosterone. LH production, like FSH production, is stimulated by GnRH from the hypothalamus. Estrogens, progesterone, and androgens inhibit GnRH production. Prolactin, pro meaning before and lac meaning milk, or PRL, works with other hormones to stimulate mammary gland development. In pregnancy and during the nursing period that follows delivery, PRL also stimulates milk production by the mammary glands. The functions of PRL in males are poorly understood, but evidence suggests that PRL helps to regulate androgen production by making interstitial cells more sensitive to LH. Prolactin production is inhibited by the neurotransmitter dopamine, also known as prolactin inhibiting hormone, or PIH. The hypothalamus also secretes several prolactin releasing factors, or PRF. Few of these have been identified, but circulating PRL stimulates PIH release and inhibits the secretion of PRF. Although PRL exerts the dominant effect on the glandular cells, normal development of the mammary glands is regulated by the interaction of several hormones: prolactin, estrogens, progesterone, glucocorticoids, pancreatic hormones, and hormones produced by the placenta cooperate in preparing the mammary glands for secretion. Milk ejection occurs only in response to oxytocin release at the posterior lobe of the pituitary gland. We'll talk more about that when we talk about functional development of the mammary glands in the next module. Growth hormone, or GH, or somatotropin, stimulates cell growth and replication by accelerating the rate of protein synthesis. Skeletal muscle cells and chondrocytes, which are cartilage cells, are primarily sensitive to GH, although virtually every tissue responds to some degree. The stimulation of growth by GH involves two mechanisms. The primary mechanism, which is indirect, is best understood. Liver cells respond to GH by synthesizing and releasing somatomatins. So these are compounds that stimulate tissue growth, or insulin-like growth factors, IGFs. These peptide hormones bind to receptors on a variety of plasma membranes. In skeletal muscle fibers, cartilage cells, and other target cells, somatomatins increase the uptake of amino acids and their incorporation into new proteins. These effects developed almost immediately after GH is released. They are particularly important after a meal when the blood contains high concentrations of glucose and amino acids. In functional terms, cells can now attain ATP easily through the aerobic metabolism of glucose, and amino acids are readily available for protein synthesis. Under these conditions, GH, acting through the somatomatins, stimulates protein synthesis and cell growth. The direct actions of GH are more selective. They tend to appear after blood glucose and amino acid concentrations have returned to normal levels. In epithelia and connective tissues, GH stimulates stem cell divisions and differentiation of daughter cells. Somatomatins then stimulate the growth of these daughter cells. In adipose tissue, GH stimulates the breakdown of stored triglycerides by adipocytes, or fat cells, which then release fatty acids into the blood. As circulating fatty acid levels rise, many tissues stop breaking down glucose to generate ATP and instead start breaking down fatty acids. This process is termed a glucose-sparing effect. In the liver, GH stimulates the breakdown of glycogen reserves by liver cells, which then release glucose into the bloodstream. Because most other tissues are now metabolizing fatty acids rather than glucose, blood glucose concentrations begin to climb to levels significantly higher than normal. The elevation of blood glucose levels by GH has been called a diabetogenetic effect. Because diabetes mellitus, an endocrine disorder, which we'll talk about later in this module, is characterized by abnormally high blood glucose concentrations. The production of GH is regulated by growth hormone releasing hormone, so GHRH, or somatocrenin, and growth hormone inhibiting hormone, GIH, or somatostatin, from the hypothalamus. Somatomatins inhibit GHRH and stimulate GHIH. Melanocyte stimulating hormone. The pars intermedia may secrete two forms of melanocyte stimulating hormone, MSH or melanotropin. As the name indicates, MSH stimulates the melanocytes of the skin, increasing their production of melanin, a brown, black, or yellow-brown pigment. Dopamine inhibits the release of MSH. Melanocyte-stimulating hormone from the pituitary gland is important in the control of skin pigmentation in fishes, amphibians, reptiles, and many mammals other than primates. In humans, MSH is produced locally within sun-exposed skin. The pars intermedia in adult humans is virtually non-functional, and the circulating blood usually does not contain MSH. However, the human pars intermedia secretes MSH in four ways. 1, during fetal development, 2, in very young children, 3, in pregnant women, and 4, in the course of some diseases. The functional significance of MSH secretion under these circumstances is not really known, but the administration of a synthetic form of MSH causes the skin to darken, so MSH has been suggested as a mean of obtaining a sunless tan, so self-tanner, if you will. So the posterior lobe of the pituitary gland, also known as the neurohypophysis, contains the axons of the hypothalamic neurons. Neurons of the supraoptic and the paraventricular nuclei manufacture antidiuretic hormone, ADH, and oxytocin, OXT, respectively. These hormones move along axons in the infundibulum to axon terminals, which end on the basement membranes of the capillaries in the posterior lobe. They travel by means of axoplasmic transport. Antidiuretic hormone, or ADH, also known as vasopressin, or VP, is released in response to a variety of stimuli, most notably a rise in the solute concentration in the blood or a fall in the blood volume or blood pressure. A rise in the solute concentration stimulates specialized neurons in the hypothalamus. These neurons are called osmoreceptors because they respond to a change in the osmotic concentration of body fluids. The osmoreceptors then stimulate the neurosecretory neurons that release ADH. I did want to take a quick minute to highlight one of the more lesser known forms of diabetes here, which is diabetes insipidus. And diabetes occurs in several forms, as I'm sure you all know. All are characterized by excessive urine production or polyuria. Most forms result from endocrine abnormalities, but physical damage to the kidneys can also cause diabetes. The two most prevalent forms are diabetes insipidus and diabetes mellitus. We'll discuss mellitus in the spotlight later on. So, diabetes insipidus generally develops because the posterior lobe of the pituitary gland no longer releases adequate amounts of ADH. Water conservation by the kidneys is impaired, and excessive amounts of water are lost in the urine. As a result, the person is constantly thirsty, but the body does not retain the fluids consumed. Mild cases of diabetes insipidus may not require treatment if fluid and electrolyte intake keep pace with the urinary loss, but in severe cases, the fluid loss can reach about 10 liters per day, and dehydration and electrolyte imbalances are fatal without treatment. So this condition can be effectively treated with desmopressin, which is a synthetic form of ADH. So that's one of the ways it differs fairly greatly from diabetes mellitus 1 and 2. All right, so back to ADH. So the primary function of ADH is to act on the kidneys to retain water and to decrease urination. With losses minimized, any water absorbed from the digestive tract will be retained, reducing the concentration of electrolytes in the extracellular fluid. In the high concentrations, ADH also causes vasoconstriction, so a narrowing of the peripheral blood vessels that help elevate blood pressure. Alcohol inhibits ADH release, which explains why people find themselves making frequent trips to the bathroom after consuming alcoholic beverages. Most of you have probably heard that something is a diuretic, which in turn, you can put that together here, has an effect more than likely on ADH. Oxytocin in women, or oxytocin meaning quick and tokos meaning childbirth, stimulates smooth muscle contractions in the walls of the uterus, promoting labor and delivery. After delivery, oxytocin promotes the ejection of milk by stimulating the contraction of myoepithelial cells around the secretory alveoli and the ducts of the mammary glands. Until the last stage of the pregnancy, the uterine smooth muscles are relatively insensitive to oxytocin, but they become more sensitive as time of delivery approaches. The trigger for normal labor and delivery is probably a sudden rise in oxytocin levels at the uterus. There's good evidence, however, that the uterus and the fetus secrete most of the oxytocin involved. Oxytocin from the posterior lobe plays only a supporting role. Oxytocin secretion and milk ejection are part of a neuroendocrine reflex called the milk letdown reflex. The normal stimulus is an infant sucking at the breast, and sensory nerves innervating at the nipples relay the information to the hypothalamus. Oxytocin is then released into circulation at the posterior lobe, and the myoepithelial cells respond by squeezing milk from the secretory alveoli into large collecting ducts. Any factor that affects the hypothalamus can modify this reflex. For example, anxiety, stress, and other factors can prevent the flow of milk even when the mammary glands are fully functional. In contrast, nursing mothers can become conditioned to associate a baby's crying with suckling, and in these women, milk letdown may begin as soon as they hear a baby cry, more of a Pavlovian effect. So the function of oxytocin in sexual activity remain uncertain, but it's known that circulating concentrations of oxytocin rise during sexual arousal and peak at orgasm in both sexes. Evidence indicates that in men, oxytocin stimulates smooth muscle contractions in the walls and ducts of the deferens, so the sperm ducts, and the prostate gland. These actions may be important in emission, which is the ejection of sperm, secretions of the prostate gland, and the secretions of other glands into the male reproductive tract before ejaculation. So studies suggest that the oxytocin released in females during intercourse may stimulate smooth muscle contractions in the uterus and vagina that promote the transport of sperm towards the uterine tubes. And we'll talk more about all of that once we get into Module 2, which is all about reproduction. One thing I do want to point out is that Figure 18-9 and Table 18-2 in the previous few slides summarize a lot of important information about the hormones of the pituitary gland. So make sure to review them carefully before considering the structure and function of other endocrine organs, which we'll talk about in the subsequent video lectures. In addition, I want you to be sure to consider some of the following questions. What are two lobes of the pituitary gland, and can you identify them? If a person were dehydrated, how would the amount of ADH released by the posterior pituitary lobe change? If a blood sample contained elevated levels of somatomatins, which pituitary hormone would you also expect to be elevated? And what effect would elevated circulating levels of cortisol, which is a steroid hormone from the adrenal cortex, have on the pituitary secretion of ACTH?